I think we can move on to the next talk, and that is by Dr. Su on retinal transplant immunology.
Okay, good morning, everyone. Thank you to the organizers for inviting me to come to give this talk. So I'm from Singapore and I'm Xinyi. So these are my financial disclosures. So in my lab, essentially, we've been looking at different strategies to treat end-stage retinal degenerative diseases. So one approach is to develop tools for surgical transplantation. Second is to understand whether we can augment the survival of these RPE cells post-transplant. And here today, I'm going to talk a little bit about a novel hypoautoimmune stem cell resource that we've been working on. And more recently, we've also gone a little bit into gene and RNA therapy. So the disease that we're interested in today is age-related macular degeneration. I think most of us are familiar that it is one of the leading causes of blindness globally, particularly in the elderly. And fortunately for us as ophthalmologists, there are multiple treatment opportunities for AMD. Particularly for dry AMD now, we've got the complementary inhibitors. For wet AMD, we have anti-VEGFs. But however, as clinicians, often many of the patients do come to us with end-stage disease, like geographic atrophy and a disciform scar, for which we currently do not have effective treatments. And this is where I think cell therapy would be a very good approach. And particularly for AMD, what we're interested in is the retinal pigment epithelial cells over there. And the RPE cells are crucial for AMD because they support the function of the photoreceptors. So some of these are as a schematic diagram showing the roles of RPE cells. Importantly, they help to turn over the photoreceptor outer segments once every 10 days. As you can imagine, they are working really hard, and because they're terminally differentiated and non-renewable, they start to accumulate DNA damage and oxidative damage over time. And as they begin to kind of be on a downward trend and they start to have dysfunction, that drives AMD. So the idea for stem cell therapy for macular degeneration is a very simple one. You get stem cells, you differentiate them into RPE cells, and then you transplant them into the eye. So here what we're thinking of now is to replace RPE cells that are non-functional with healthy RPE cells and hopefully be able to recover some of the vision. So this is not a new idea. There are multiple high-profile publications that have been out there in the US and the UK, and particularly they are looking at using autologous cells. So here meaning they take cells from the patients, they reprogram them into RPE cells, and then they inject back to the same patients. This will help to overcome immune rejection. Alternatively, many of the groups have also looked at using human embryonic cells. And I think more encouraging is that more recently there is a clinical trial by Lineage Cell Therapeutics indeed saying and showing that transplantation of embryonic-derived RPE cells can indeed improve vision as well as regain some of the anatomical structures. However, there are still remaining challenges to bring this into clinical reality. I think one of which I alluded to earlier is the optimal surgical approach. Do we do cell suspension? Do we do monolayer transplant? I think very importantly also is to develop an ideal cell resource. So for embryonic-derived stem cells, it's a limited resource and potentially ethical concerns because we're deriving it from an embryo. For induced pluripotent stem cells, there are two large categories. One is autologous, which I alluded to earlier, where you take cells from your own patient, reprogram them, and inject it back. And you can imagine this is a very laborious process where you need to develop one particular cell line for each patient. And these cell lines would have the inherent AMD risk alleles that arising from these patients. The other approach is to use Allogeneic. Basically, you have a master cell bank, and then you use that to treat patients in a broad manner. However, with that, you have the potential problem of immunogenicity. So you need to do some form of immune suppression as you would do for any sort of organ transplant. And also, more importantly, people are also thinking about we can get the stem cells in, but are they functioning? Do they make the connections? How can we enhance survival and integration of these post-transplanted RPE cells? And this is the topic of our discussion today. So in our lab, we've been working very closely with a biotech company called Cell Research Corp in Singapore. Here, our hypothesis is that if we were to derive stem cells from the cord lining cells, and because pregnancy is an immune miracle, this maternal-fetal interface is immune privileged, and if we get cells from this epithelial cells of the umbilical cord lining cells, perhaps we don't have to use immune suppression as much. And the further advantage of CLIPS is that it's unlimited, so umbilical cords are being thrown away every day, so it's a resource that we can always come to. They are very young cells. They age 0, genetically, somatically pristine, and they have HLA, so we can HLA match them as well. So the idea is that this potential cell source could be a universal cell source that we could use, overcoming the need to use autologous approaches. So the first experiments we did was in vitro. So we have the cord lining cells, CLECs. We reprogrammed them into IPS, so we call them clips, and then we differentiate them into RPE cells. And you can see on the top here, the clip cells, sorry, it doesn't project very well, sorry. So basically, in the parental lines, they have lower expression of MHC class 1 and MHC class 2. And after we differentiate them into RPE cells and we stimulate them with interferon gamma, we also see that there is reduction in expression of MHC class 1 and 2 compared to the skin-derived and ES-derived cells. And next thing we did was then mix them together in a lymphocyte co-culture. So we mix RPE cells with T cells. Consistently, you can also see that the RPE cells have, they don't stimulate proliferation of both the CD4 cells as well as the CD8 cells. So now we moved on to an in vivo system. Here we are using mice that are humanized, meaning mice with a human immune system. We injected the cells of RPE-derived, ES cells, clips as well as skin-derived, and we look at both a systemic immune response as well as a localized response. So here, in the subcutaneous space, when we explanted the transplants, you can see that the pigmented RPE cells still remain. And then we also look at the serum and measured pro-inflammatory cytokines. Here again, you can see that in purple, for the clips-derived RPE cells, there was a reduction, there was less induction of pro-inflammatory cytokines as well as reduction in T cell activation in the various organs. And we then looked at the localized areas. Here we are looking at human CD45 cells stained in red. So this is a pan-immune cell marker. You can observe here that in the clips on the extreme left-hand side that there was very little infiltration of CD45 cells compared to the ES-derived and the skin-derived RPE transplants. Then we leverage on single-cell technology to kind of understand what are some of the processes that are happening at the cellular level. What we did here was explant the cells and did single-cell transcriptomics. In purple, you can see again, comparing the pre-transplanted to the post-transplanted RPE cells, there was a reduction in upregulation of the MHC class 1 and class 2 compared to the skin-derived cells. And next, we wanted to again understand or characterize the immune cell infiltration in the microenvironment post-transplantation. And here you can see in purple again for clips, very interestingly, there was a huge population of B cells and myeloid cells compared to the skin derived in the brown and the green, and there was a much less population of T cells. So this again is a very unique immune microenvironment that we observe specifically for the clip cells. Then we dug a little bit deeper to say, what is the population of the T cells that are available there? And you can see in the circle over there that very interestingly for clips, there was a predominance of Tregs. So Tregs is a subpopulation of T cells that have a kind of a hypoimmune property. They regulate other T cells by secreting anti-inflammatory cytokines and then downregulates the immune response. So again, this was very unique to just the clips RPE cells. And now we are undergoing further analysis to identify immune modulatory factors. And our hypothesis is that the clips RPE cells somehow secrete factors to upregulate the surrounding Treg cells and therefore suppress the immune response in the microenvironment. That was in the subcutaneous space of mice. So we wanted to ask the question, what happens in the subretinal space? So here we use a larger animal in rabbits. We transplanted the monolayers of the clip cells into the rabbit eyes, and we did some phenotypic analysis over there, essentially showing that consistently in the clips RPE cells, the transplants were quite well tolerated. Similarly, we also did single-cell RNA-seq, so we explanted them, compared the in vivo cells to the in vitro cells. And again, the clip cells do behave very differently. Again, in purple over there, we saw that they have a higher expression of mature RPE markers. So our hypothesis is that because it's transplanted, there's less of an immune response. These RPE cells can then flourish and act as RPE cells. So we looked at gene expression in terms of visual cycle proteins, extracellular structure organization, melanin synthesis, and phagocytosis. Again, you can see in the red arrows there, transplanted clip RPE cells are in a way like super RPE cells that can outperform the other RPE cells. And consistently, we saw there was a downregulation of MHC class 1 and 2, which is what we observed already in the in vitro cells, as well as the humanized mouse models. And interestingly, we saw an upregulation of TGF-beta pathway, which we postulate could be one of the mechanisms in which these cells are different from others. So last of all, we went to a non-human primate model. So here we transplanted CLIPS RPE cells, compared the skin RPE cells into non-human primate in the macular area without without immunosuppression. So this is a xenograft. And at 3 months, you can see in the yellow arrows, the CLIPS RPE cells were better tolerated compared to the skin RPE cells. And we have followed this up for up to a year in some of our models, and also have done functional studies, which I will not show here. So in summary, CLIPS RPE cells is potentially a universal source of hypoimmunogenic cells. We've shown that in vitro. We've also showed you some allograft data in a humanized mouse model. We looked at localized immune response, systemic immune response. We have some understanding of the mechanisms, but we are still trying to uncover that, and also shown you some evidence of transplantation into large animal models. And we have filed a provisional patent application for this, and hopefully in the near future, we'll be able to bring it to clinical trials. So I'd like to acknowledge my team, my funding body, and I'll be happy to take any questions. Thank you.
Questions?
Very nice talk. I often have patients tell me that, oh, we saved cord blood after our child's birth. And they say, oh, is there something we can do with it? And I say, I don't really know. What they save, could that be used to make these CLIPS cells, or are they just saving, blood cells, or do you get epithelial cells?
Yeah, I think it very much depends on the package that you get. So I've stored my own cells as well, and with a Singapore company, and they give you the option of just saving the cord blood itself or the epithelial cells. So it's very much dependent on your service provider. But what we've also observed is not the same across all cord lining cells. We do see some super performers. And of course, what we've shown you here are the high performers. There is a huge variation across different cord lining cells that we deal with. So I would think that, we couldn't use the same patient's cord lining cells and assume that they will function as well.
Yeah. Questions? Anybody? Okay, I just have, one query here, that you have actually now manipulated the immune system, and so to an extent the immune response has been, taken care of.
Yeah.
But what about, we're talking about transplanting these cell sheets into dry AMD patients, and there's a milieu of cell death and degeneration there. So if you put these cells into that environment, what would be, it's not an immune-related issue, but generally, so what would be I don't know whether you have any such experience, how long you expect the cells to survive, because, you have that sort of in the recipient.
Yes, yeah. Thank you very much for your question. So this is exactly one of the things we would like to look into. So we have developed disease models in monkeys. So we're using micropulse to induce localized RPE dysfunction, and that will partially disrupt the Bruch's membrane also. So I think we will hope to kind of challenge it a little bit more to see if the transplants in a more immune complex situation, do we still get that same effect? But this is certainly something that we can look into.
Okay, any other questions? Okay, yeah.
Were they HLA mismatched samples?
Yes, they were HLA mismatched because we wanted to push the system a little bit more.
Okay.
Yeah. Great, thank you.